So when we finished up lecture last week, we had gone back over action potentials and how myelin on the axon helps to propagate those action potentials down the axon through saltatory conduction so that the action potential can jump from node of Ranvier to node of Ranvier, terminating in our axon terminals. And so once the action potential is generated from the receptive ends, the dendrites of the soma, the axon cell body, culminates to threshold at the axon hillock, and then fires down the axon through saltatory conduction to the axon terminals, then we go into our synapses. And we have discussed neuronal synapses before with the neuromuscular junction. So hopefully this will all be a review. But a unique feature of the nervous system is the fact that we also have electrical synapses in the nervous system. You can have many different synapses with an axon. You can have axon dendritic synapses, so that the axon synapses with a dendrite. Axon somatic. Remember, I've told you that the cell body is sometimes referred to as the soma. And so somatic is when the axon actually forms the synapses with the neuronal cell body. And you can have axonic synapses where it fuses or forms a synaptic junction with the neuron axon. With each of these, we have our presynaptic neuron, the one that is sending the signals, and the postsynaptic neuron, which is receiving the signals. And then that transmission of a signal can be chemical via our neurotransmitters. When we looked at the neuromuscular junction, that was acetylcholine. Or it can be an electrical signal. This allows for voluntary movement, cognition, sensation. On average, each presynaptic neuron synapses with about 1,000 postsynaptic neurons. And a postsynaptic neuron can have as many as 10,000 presynaptic connections with different neurons. So a lot of communication between neurons within our nervous system. With an electrical synapse, these are coupled via gap junctions, so they are actually physically linked. Remember, with our chemical synapses, we always have that gap between the axon terminal and the postsynaptic neuron, or it can be a gland or a muscle. And so there's always that slight gap or synaptic cleft between the chemical synapses. But with the electrical synapses, they're actually physically connected via gap junctions, so that so they're still not touching, but they are connected via the gap junctions. And those gap junctions will allow ions or other small substances to flow, which allows a more rapid transduction of the action potential from the presynaptic to the postsynaptic. Another unique feature of our electrical synapses is it can be bidirectional, whereas our chemical synapses is only unidirectional. So chemical synapses can only flow in one direction, but with electrical synapses, it can flow back and forth between the presynaptic and postsynaptic. And as I just mentioned, it's nearly instantaneous. Only delay is it takes the presynaptic neuron to depolarize, so less than 0.1 milliseconds. Whereas a chemical synapses can be 1 or more milliseconds because it has to wait for the neurotransmitter to diffuse across that entire synaptic cleft. And so here is a visualization of our electrical synapses and how the gap junctions form an open pathway between the presynaptic and postsynaptic neurons. Chemical synapses actually make up the majority of the synapses in the nervous system, more efficient than electrical synapses. because they convert electrical signals into chemical signals, so no signal strength is lost, and so it is a much more efficient synapse. So again, our electrical and chemical synapses compared. 3 structural differences. Synaptic vesicles are only in our chemical synapses with the chemical messengers. Our synaptic cleft, extracellular fluid filled space, separates presynaptic and postsynaptic neurons found in chemical synapses, but our gap junctions connect the neurons in the electrical synapses. And so here is a chemical synapses. I actually have that synaptic cleft where the presynaptic and postsynaptic never truly touch. And again, this is just like our neuromuscular junction where the axon synaptic vesicles fuse with the presynaptic membrane, release their neurotransmitter into the synaptic cleft, it diffuses across, adheres to our ligand-gated channels, usually opening sodium channels. Sodium flows in, depolarizes, does a local depolarization of this postsynaptic membrane, and then that depolarization is carried into the postsynaptic receptor. With their chemical synapses, we have that delay because the neurotransmitter has to diffuse across, and you have to have a neurotransmitter receptor for that particular neurotransmitter. Again, as we pointed out earlier, chemical synapses are unidirectional, whereas electrical are bidirectional, but they allow for a viable signal intensity. So electrical do not have variable sensitivities, variable signal intensities, whereas our chemical synapses can. So it can regulate via the amount of neurotransmitter released. More neurotransmitter, more response, a stronger response in that postsynaptic neuron. So remember our events at our chemical synapses that I just mentioned. So action potential arises, stimulates the synaptic vesicles to fuse with the presynaptic membrane, releasing the neurotransmitter into the synaptic cleft, binding to our ligand-gated channels that usually lead to opening of a sodium channel is a common one. And if it reaches threshold, if that local potential reaches threshold, then causing an action potential to be generated. And here's your big picture animation again. Remember, you can find these in my study area.
Neurons receive stimuli from other neurons or from sensory stimuli in the internal or external environments. If the incoming stimuli are significant enough, they depolarize the neuron's trigger zone to threshold, and an action potential is generated. The action potential is quickly propagated down the axon and reaches the axon terminals. At this point, synaptic transmission occurs and allows the neuron to communicate with other cells, including other neurons, muscle cells, or secretory cells inside glands. The location of communication between the neuron and its target cell is called the synapse. The most common type of synapse in the nervous system is the chemical synapse. As the name suggests, these synapses use a chemical, known as a neurotransmitter, to transmit the signal from the initiating presynaptic neuron to the receiving postsynaptic neuron. Over 100 neurotransmitters are used by our nervous system. Examples include acetylcholine, dopamine, norepinephrine, and serotonin. Remember that there are 3 parts to a chemical synapse. First is the axon terminal of the presynaptic neuron, which contains synaptic vesicles that store neurotransmitters. Second is the postsynaptic neuron, whose membrane contains neurotransmitter receptors. And third is the microscopic gap between the two called the synaptic cleft. Let's now look at the events that happen at a chemical synapse between two neurons. First, an action potential arrives at the axon terminal of the presynaptic neuron. The resulting depolarization in the axolemma opens voltage-gated calcium ion channels. Extracellular calcium ions enter the axon terminal following their electrochemical gradient. This causes synaptic vesicles to fuse with the presynaptic membrane. The vesicles then release neurotransmitters into the synaptic cleft, a process called exocytosis. Next, the neurotransmitter molecules diffuse across the synaptic cleft and bind to receptors in the postsynaptic membrane. These receptors can be ionotropic, meaning the binding of a neurotransmitter directly opens an ion channel, as seen here, or metabotropic, meaning that it opens ion channels indirectly. The resulting movement of ions across the plasma membrane will cause a change in the membrane potential of the postsynaptic cell. The average neuron can form synapses with about 1,000 other neurons, thus forming a complex neural network. Chemical synapses provide the means for transmitting signals within that neural network. The strength of the signal varies depending on the amount of neurotransmitters released and the frequency of stimulation. The effect on the postsynaptic cell depends on the types of receptors present in the postsynaptic membrane.
All right, so let's do a quick review. The correct order of events at a chemical synapse is, I'll give you about 30 seconds to choose your answer, see if you're right. All right, what is it? It's C. Remember, an action potential is the first thing that happens. So you can read through these and immediately see, all right, action potential arrives at the axon terminus. That's the first one. This is the only one that's got that. So it's the only one that can really be correct. Action potential arrival at the presynaptic terminus causes opening of voltage-gated calcium channels. So that action potential arriving at the synaptic terminus, so on the axon, the presynaptic opens the calcium channels to cause the synaptic vesicles to fuse with the membrane and release its neurotransmitter. Then when that neurotransmitter is released from the vesicles of the synaptic terminus, results from an influx of calcium into that synaptic terminus. Neurotransmitter binding to the receptors of the postsynaptic membrane cause what? And this is going to be a little different than the neuromuscular junction because it can be an inhibitory or an excitatory signal. So it opens or closes ligand-gated sodium channels. So it can stop an action potential or it can begin an action potential because the nervous system has both excitatory and inhibitory signals, as we talked about last week and as we'll look at in the next video. And so those postsynaptic potentials we'll get into in the next video. So continue on and I will see you in the next one. Have a great day.